Bally-Cuif. I mean, you're research director at CNRS, and you're going to move your lab to the Pasteur Institute, which is the epitome of excellence in this country, as we all know. And you are interested in regeneration and neurogenesis in the fish. Well, they are not supposed to be the smartest animals on Earth, so what brought you to studying fishes?
So, adult neurogenesis, in fact, has been found in all vertebrate species that have been looked at, mammals, but also birds and reptiles and amphibians and fish. And it turns out that fish are really, compared to other vertebrates, champions of adult neurogenesis. So they maintain many neural stem cells in their adult brain, and they can use these neural stem cells for producing neurons continuously through life, and also for very efficient regeneration. So these are very interesting animal models to compare with mammals in particular, to try and understand why neurogenesis is not very active in mammals and why it's very active in fish. So we use these animals to try and define the molecular pathways in particular that are different between the different vertebrate species, and eventually, you know, maybe in many years, use what we learn from fish to try and boost adult neurogenesis in mammals. So it's not a matter of intelligence, it's really the basic processes of neural stem cell biology are conserved between zebrafish and mammals, and we try and understand why the efficiency of the process is higher in fish than in mammals.
But if you make new neurons all the time, I mean, how can you remember anything? You know, you should be totally stupid if you make too much neurogenesis now.
So there's been studies in mammals that indeed show that the making of new neurons is important for forgetting some past episodes. You know, it's very difficult to measure intelligence in an animal, so you can measure learning and memory, and this has been done in fish, and they in fact are capable of learning and they are capable of remembering over long periods of time. Studying why adult neurogenesis could be useful or you know, less useful for this is another topic really. So what we are interested in is really to try and understand, and where I think the fish can bring something is to try and understand what is making the neural stem cells and the adult neurogenesis process efficient or less efficient. In many different contexts in the brain.
But has neurogenesis anything to do with the fact that if you remove a piece of brain in a fish, it just regrows, which never happens in the human, right?
This is true. So if you make a lesion, a mechanical lesion, for example, in a fish, the fish is capable of repairing this lesion by the production of new neurons within just a bit more than a week. And this is by recruiting neural stem cells that are present in the fish brain and that can be stimulated and brought to the completion of making an efficient neuron. What happens in mammals, usually when you make a mechanical lesion, so this has been very precisely compared, is that you can also boost your neural stem cells in the mammal, but they will not make it through the entire neurogenesis process. So this will abort somehow. They will make either neurons that will die or they will not complete the neurogenesis process. So it's very interesting to compare then both species to try and understand why neurogenesis can be used for repair completely in a fish and not in a mouse. And the current set of data that is available at the moment suggests that the difference is not in the stem cells themselves, but is in the environment that the stem cells encounter when they get boosted and when they produce their neurons. So the environment is very strongly permissive for neurogenesis in fish and is not permissive in mammals. And there's a lot of work currently conducted to understand what this permissiveness or non-permissiveness means.
So do we have any hint of what's going on? I mean,
so the major hypothesis at the moment on this front is that what is responsible for blocking the completion of neurogenesis in a regenerative context in mammals is the inflammation reaction. So inflammation that follows the mechanical lesion is a process that is initially necessary to boost the stem cells, but later on in mammals is sustained over a long period and stops the neurogenesis process. So we are trying to understand at the moment what is different in the inflammation reaction.
In fishes?
There is inflammation, there is a first phase of inflammation. But this very rapidly stops. So this first phase is believed to produce, secreted factors and molecules that will boost the stem cells. But once the stem cells are boosted, this inflammation reaction, in fact, if you sustain it, counteracts the neurogenesis process. And in fish, the inflammation reaction stops very soon, while in mammals it perdures. And it's what is the hypothesis at the moment in the field is that there may be different inflammatory molecules in the two species, and this is an active domain of research.
And what is the link between neurogenesis and regeneration? I mean, is there anything, if you cut a brain or cut an axon or whatever, it will regrow in a fish, it will hardly regrow in a mammal. And has this anything to do in common with neurogenesis, or is it a totally different mechanism?
The regeneration goes via the stimulation of the endogenous neural stem cells. This is true both in a fish and in a mammal. In both species, you have areas of the brain that have active stem cells and areas of the brain that don't have active stem cells. It's just that between fish and mammals, you have more areas with active stem cells in a fish than in mammals. But both have both categories of brain territories that are neurogenic or not neurogenic under physiological conditions. If you make a lesion, you will be able to reactivate latent progenitor cells in both species, in both types of territories. So the fact that you can regenerate is not really directly linked with whether or not neurogenesis is active under physiological conditions. It's really linked with the fact that the environment is permissive in one species for the completion of the neurogenesis process and is not permissive in other species like mammals.
Okay, so there is hope that we shall soon have drugs allowing us to regenerate our brains and maybe our heart, because I believe it's more or less similar. You have the same problem for all organs.
This is correct, and in fact the molecular mechanisms that happen to be involved in activating stem cells are often shared between the different organs in the mature body. Now, are we close to a way of boosting neurogenesis in conditions that necessitate repair, for example, upon lesion or upon neurodegeneration? Or stroke? This is another issue. I think we have genetic means in animal models for boosting neurogenesis. It's more complicated to apply now these genetic means to the human situation, but it's efficient in mammals, in particular, in rodent models in particular.
Well, let's hope this will happen before it's too late for many others. Thank you very much.
Thank you.